Today we're talking to José Luis Ricon about aging and what we can do about it. He's the head of theory at Retro Biosciences. We thought it would be really good to have one of our early episodes be on theories of aging. Obviously, you have expertise in this area.
Mm.
What do you consider the goal with respect to aging, health, and lifespan?
Yeah, I think in the aging field, I'd say it's similar to broadly healthcare or biomedicine. It's almost like a theological value. There is value laden. You're making a value statement about what's good. We're saying that health is good and that the aim is to increase it, and biomedicine broadly. In the case of aging, the ultimate goal is progressively increases in healthy lifespan. And so that doesn't have a ceiling. You could keep going and going. From the point of view of aging, you could say that we have gotten increases in healthy lifespan through various means. Historically, reduction in infectious disease, prosthetics, all sorts of stuff. The aging differentiation might be the dual thesis that increases in healthy lifespan, will be achieved through aging-related mechanisms, and that they can only be achieved through aging-related mechanisms. That, like, once you addressed low-hanging fruit, you are left with systemic degradation of everything. Basically, there is no approach where you do one molecule at a time. The POC kinase doesn't work anymore, and you have to do aging. So ultimately, all medicines become aging medicine at some point.
Yeah, and that actually leads into something I was going to switch to anyways, which is there's a lot of theories and paradigms for how to think about aging. It's tended to go through these cycles where people get really excited about a particular discovery, whether it's telomeres or senescent cells, and gets really hyped up. And then it always turns out like, oh, it's more complicated than that. Maybe tell us a little about your story because you're relatively new to the aging field. So when you started reading about this and when you started reading about theories, what was your path to learning and what did you think about the different theories you came across and what ultimately do you consider your own framework for how you think of your own theory of what aging is?
I guess when I first got into aging, or rather, the first time I was exposed to ideas in the field was in college through Over the Gray's book, Ending Aging. Although at the point, I didn't pursue it much longer. I thought, cool, and then I moved on. So I had that sense framework of aging in mind, which seemed reasonable. You basically parcel it out into some like roughly like disjoint sets of kind of damage, and then you say, well, if you address all of this, that's it. Theories of aging are kinds of theories that they are not the same. Like, for example, there are theories of like why is aging there and why do some species age and other not. This is where people will point to things like group selection or lack thereof, or they will point to antagonistic pleiotropy in how things came to be. There's a question to be had about, no, there's a line that goes, nothing makes sense except in the light of evolution. I don't fully buy that. I think you can fix something without knowing why it's there, knowing why it might help, but you don't need that necessarily to change something. And then you have theories about like how does it work? So you take a human being or take a worm or take a mouse. It's like, well, this mouse is aging. What's happening? There you get to theories. One of the earlier ones, the Harman like Ross theory. This is where we got the reactive oxygen species. This is oxidation, and where the antioxidant craze came from. It's like maybe there are these metabolic byproducts causing damage through this Ross. If we address this, we can address aging. Some interesting studies in vitro showing that, sure, if you induce Ross in cells, they'll hate that. I think it was similar to the telomere theory. This is back in the day where people were like, well, it seems like cells stop growing after a while. This sounds like aging. And why do they stop growing telomeres? It was not maybe unreasonable to believe back then that if you just elongate the telomeres, that's it. Your cells keep growing and they don't senesce and they live forever. The German Corporation was founded on this thesis. I wrote a whole article about telomeres. I guess initially we had the UR theory or the classic GF1, mTOR. This is what I like to call aging 1.0, what has been in the field since India Canyon and even before that. But so there are theories that claim to be comprehensive, where they say aging is driven by one thing, and maybe these two examples being telomeres and Ross. And there are theories that are more like, here's one driver of aging, and then it's like, okay, cool. The telomeres, and I guess that eventually led to the hallmarks of aging paper, where they say, well, there are these 11 things. It doesn't say how they are connected necessarily or what the hierarchy is and so on. But then you could say, well, okay, so there are these frameworks. I think there is one paper that I was looking the other day that had like 100 or 1,000 different theories of aging. To me, the what a theory of aging should have, at least for a useful one, should say, well, what is this aging that you're speaking of? Defines like what's the thing you're going to be pointing at, and what's driving it, what goes from non-aging to aging, and then what happens inside the black box, because that then points you to where you, because ultimately, because the objective is to address this, you want something that tells you where to intervene, not just something that tells you here there is. And it's like, okay, cool, so what? Even the people that say that there's there's the aging is programmed view, where aging is there are two versions of this. One is that we evolve to age, which is the most controversial one, and then there's the I guess more like it's a runaway developmental program. You could say, well, so what? How does that imply addressing it? It's not like they imply you cannot reverse those programs. Some of these people will say reprogramming shows this is actually correct. It also shows we can reverse it. So I think that's what a theory should have. It's like what is aging? What are the drivers? What is the causal structure linking those causes to the downstream aging phenotypes? If you like, let's say, list down what is it to be an old X individual, and this has to be done by species. The way in which a worm ages is not quite the same in which a human ages. There are some similarities, or even you could argue that the way a modern human ages is different from the way a human would age in a different environment. Like you could, for example, find an intervention that improves health and lifespan in a context where there are, for example, no viruses, but it reduces lifespan in other contexts where there are viruses, for example. So in which environment is this being done for? That's how I tend to think about it. And I can go deeper into what goes in these boxes of aging and the causal structure and so on.
Yeah, well, you've brought up a ton of different topics that are all worth digging into, actually. I think one of the ones that you mentioned earlier was in the gerontological literature. Historically, there's a lot of cases where they would increase ROS and show increased rate of aging. The problem is that there's a lot of ways you can create a progeroid model that don't actually tell you much about aging itself, right? Breaking something is a lot easier than improving on something. So there's a lot of ways you can generate a progeroid model that doesn't actually, but then fixing it doesn't actually imply much for normal aging. I think that's like historically one of the issues.
One other thing is like in the hallmarks of aging, even the original paper, the way they define it is like you modify this one lever or this one knob and it accelerates, or you turn it down and it ameliorates. But often in the details, they just use lifespan as the metric for aging, right? So then it's not clear what we're talking about when we're talking about aging. Some researchers mean it very clearly there as mortality increase or exponential mortality increase, like the Gompertz kind of aging. But then there's other people who think aging more is like the phenotypes, or maybe it's a combination of both.
So something I like to point out with the hallmarks of aging, I did the paper. Yes, a hallmark is something that if you increase it or play with it, you are increasing or decreasing the rate of aging or lifespan. You could say in humans, it's LDL cholesterol or general ApoB particles. Is that a hallmark of aging because it goes down with age? If you pump someone with cholesterol, they will get heart disease and they will die. If you clean cholesterol from someone, they will probably live a bit longer because you're not having any heart disease anymore. It's because of death. And it's very narrow. But you could say, on the one hand, it seems like it's important in some way, but in another way, there's some other part of your brain goes like, this doesn't seem like an aging thing. It's so narrow, right? In aging, there's this idea of shared mechanisms, or looking at things in which all cells are similar and studying the breakdown of those. Every single cell type has to make mRNA, make proteins, recycle proteins, and so on. The breakdown in this is similar. It's the ways in which these processes work is the same across cell types. Breaking ribosome or mitochondria working worse can happen in every cell type. Studying this throughout all tissues has been core to aging, but there's also tissue-specific aging. For example, in the blood vessels, you have this cholesterol problem that's not present necessarily elsewhere. And so maybe in aging, people tend to jump into thinking about cellular aging. The hallmarks of aging are not fully cellular aging, but they are kind of cellular aging to some extent, or think of cells. But then once you move away from just the cells and look at the whole organism, there are things that are not just cellular aging, that are also real, and you may not, probably if you even fixed all cellular aging, you would not fix those necessarily. I think the latest ones, people are adding like extracellular matrix degradation or stiffening as another one, which is not quite cell. We don't know yet with high confidence whether cellular rejuvenation will also be able to reverse the ECM. There are these debates about can cells break crosslinks if they're young enough or start over too slow. But it might be that they cannot. And in that case, you have to address non-cellular aging as well.
So I want to get right back to the original question. The question is, how complex is this problem? One of the things that we want to try to get people's view on. In the worst case scenario, it's literally tens to hundreds of thousands of metabolic processes that are drifting away from homeostasis almost independently. But as they get worse, they're making each other worse too. You get this exponential process, and there is no linear cause and effect, right? Like it's actually all just a network where
Yeah.
everything's affecting everything else. That's the worst case scenario. Some people, especially in the cellular reprogramming field, make strong cases that there are particular drivers, like maybe it's epigenetics or it's the ECM. So where do you personally fall? Do you have a personal view right now, or are you more agnostic right now?
I do have a view which will not surprise anyone given where I work. But yes, it is indeed true that the thing with aging is that it's not that one gene is broken or that it's like classically in medicine you're like, well, there's this one precise target and we want to like go hard on that, but it's kind of really like taking an old horse and kicking it to make it run. At some point the horse just dies. It's like you just cannot, like, by means of the molecules, reverse the brokenness of the entire thing. Or for that matter, gene editing, you cannot. It's not a genetic problem. All genomes are valid. Most people when they are young, no matter what mutations they have, even people that have early onset Alzheimer's mutations, are mostly okay when they are younger. So I think most genomes are valid. The genome is okay, then what's the problem? So I do think there's a lot to be said for it's the epigenome. The epigenome is driving a lot of it. Now, the why. So why this? So there are, I guess, some things to be said here. One is in vitro, there is no other intervention that can comprehensively reverse all damages we can measure in vitro. As I said, aging is already solved in vitro. I wrote a blog post on this looking at the evidence. This seems to indicate something quite interesting. If one bodies evidence, it just doesn't immediately implicate or imply that that's the only way of doing that, but they have not seen other ways of doing it. Another way one could think of about like where the big levers would be if you poke at one of them, how strong and permanent is the effect. For example, there's a paper from Ken Rush and Horvath that was presented at this Jersey conference 2 years ago. It's already published. And what they do is measure, they poke at, they do reprogramming, I think, or sorry, they have fibroblasts and then they apply various manipulations to various hallmarks and they measure the impacts on others. Rapamycin, for example, slows down epigenetic aging as measured by DNA methylation clocks in vitro. It doesn't revert it, but it slows down. What things do we know that slow down genetic aging? Not many. Reprogramming is the one that or partial reprogramming can do that. The paper isn't exhaustive. One could do a more exhaustive experiment and say, okay, well, here's one hypothesis. Suppose you upregulate autophagy substantially, and then the cell can catch up in its cleaning process, like get fresh DNA repair machinery, and all the things are now like up to speed, all the damage is clear, and maybe the cell can get some rejuvenation from that. I think the reason why I think epigenetic aging is kind of privileged, and it's probably like the thing at the cellular level. Cellular aging is not organismal aging, but I think it's that we're made of cells. It has something to do with it. Is that what's happening with the epigenome is that you're losing information about what cell type you're supposed to be. This information is not backed up somewhere. The cell doesn't know how to be a fibroblast. Once you lose methylation in one area, it's like, oh, I need to put all these 10,000 methylation sites again. The cells have programs to blank the slate and then to become something again. But these things don't really turn on just naturally unless you force them to through programming. So unless we have a way to recover this information through other way, it's hard for me to see how, without reprogramming, you could actually rejuvenate in situ or ex vivo. You could also rejuvenate by replacement. You could do something else altogether. But, like, for example, if you want to make your neurons young, given the current state of our knowledge, I think that reprogramming has to be part of a solution. It's great that it is, because cells have this program in them already. We don't have to engineer an entire pathway for comprehensive rejuvenation. Do you mind how hard would it be to do all the epigenetic edits one by one in the entire cell to fix a cell? Instead, we can just press a program and then boom. That's quite nice. I mean, it's still early days for reprogramming, but it's very satisfying that this is already there.
Yeah, you brought up the point about the lack of a reference, right? The individual cell doesn't have a reference it can refer to. Although there was a generative algorithm during development that produced that cell type in that state. That's why, in theory, reprogramming could work, because you are reliant on the genome itself. But then that brings up nuclear DNA mutations and clonal amplifications of different mutations and things like that, which you can't fix through any kind of epigenetic reprogramming.
You would have to do edits in that case. That's, to me, the final frontier. One of the final frontiers is DNA mutations, because you just have to edit them. There are potential workarounds. Like, to go more into one of the things we're doing at Retro, we could say, well, as people might know, we're pursuing, and I'm not like dumping this line here because I have to shield Retro, but we're doing reprogramming at Retro, and we're doing in vivo and ex vivo, and the ex vivo programs are easier to do because you can do anything you want to cells ex vivo, and you can go very hard on reprogramming. And if something bad happens, it doesn't matter. You can sort the cells and pick the good ones, make sure there are no iPSCs. But we can make hematopoietic stem cells young and then fully rejuvenate all your blood cells, except T cells, because, no thymus when you're old. But then it's like, yeah, but what if those HSCs have already been pre-selected through those years to be pro-leukemic or something like that? In that case, you still have the option of like sorting HSCs and pick the less mutated ones, or like edit them before putting them back in. We might not do this for the first generation product. We may instead choose patients that our therapy is compatible with, or you could say it's like benefits and risks. Even if you're increasing leukemia risk by a little bit, and you're getting these other lifespan benefits, it might be worth it. And then treating leukemia is amongst it's with melanoma is the easiest cancer to treat, so it's not that bad. But it is bad. This has been studied a lot in HSCs with people who talk about like clonal hematopoiesis of indeterminate potential or CHIP. But it happens elsewhere. In every tissue, you have the generalized cancer principle. What can reproduce the most shows up more and it keeps spreading. Fibroblasts, the liver, the gut epithelium. So you have these effects. But the question is this rate limiting? It goes back to what we talked about earlier. If you break all the antioxidant enzymes in a mouse, the mouse probably will live less. But that's not what happens. It's a form of aging that's not really realistic. One could say, well, if we don't think yet about mutations, how much can you push it? Similarly with telomeres, telomeres have had their hype cycle, but that doesn't mean that they do not matter at all for aging. It's like if you live long enough, sure, your high turnover organs the telomeres will shorten, you will stop having replication, so they will affect you in some way. But the question is, does it affect you? Does it matter like here and now? Probably not in humans. So that's something that we think about at Retro. What are the limiting steps? Because it's one thing to think about how to get the pie in the sky, the unlimited lifespan, healthy lifespan, and then there is what can we do here now fast? What can we get in the FDA in a few years? What's the first step to it? People talk about longevity, escape velocity. What are the things you can begin to do right now without having to figure out everything?
So mentioning longevity, escape velocity, that's a good segue to, I think, what we usually would ask earlier in the podcast before we got into the weeds on all these things. But we are talking about building a roadmap to solve aging. And so what is your personal roadmap? Like longevity escape velocity is this concept that has obviously been discussed, and I think it has its pros and cons. There are some negatives to thinking about it in those terms that we could talk about later. But before we bias you at all, what's your personal thinking about like what's the roadmap? How do we get from where we are now to aging being a solved problem?
I guess there might be two answers to that. One would be my own thoughts, and then which are like coupled with my employer's thoughts, which are not necessarily the same, but they are tightly connected. If not, I would not be working here. There are various ways to address aging. I mean, I, for example, think that I guess one could be, okay, well, what are like low-hanging fruits that get you lifespan in some way? And you could say, okay, well, for some people, Ozempic will be such drug. It's for other people, PCSK9 inhibitors will extend the lifespan by virtue of like getting rid of heart disease but low in cholesterol. That's an easy win. Maybe rapamycin or like rapalogs will also work, but you get this whole local optimum. It doesn't go beyond that. Some people get really worked out about rapamycin. I'm like, it doesn't go anywhere. You cannot knock out mTOR and then expect to live much longer. You need it. So then I say, okay, what are the potential high upside, even if unknown areas? And reprogramming comes to mind. So if you wanted to make an old organism into a young one or a human, how could you do that? One is you could do it in situ. You can do it by replacement, or rather by taking cells out and putting them back in. And you could in principle do it in an autologous, so in an allogeneic way, that is with other person's cells, but then you have to immunosuppress. And even though there are people trying to build stealthy immune cells, universal iPSCs, I'm not a huge fan. To simplify, let's say it's in situ, it's ex vivo for your own cells, or whole cell replacement. This is where you would grow an artificial liver and then put it in, and now you have a young liver and things like that. Of course,
you can't do tolerization through bone marrow transplantation in conjunction with as well.
You could also do that, but then you are left in this you have this chimeric organism, and you have to do both. Yeah, so Retro is focusing right now on autologous in that sense. You get your own cells, whatever good or bad they were, you get them, you're rejuvenated. In your roadmap, you have mentioned, for example, cryonics or cryostasis. That to me, it's a parallel track. It's more like delaying as opposed to addressing the problem. It's buying more time to address the problem as opposed to just addressing it. One could also say, instead of fixing biology, you could have a Zyborg solution where instead of having, let's say, how do you fix osteoarthritis, you instead get a joint replacement. And now you don't have to worry about osteoarthritis because you have a titanium joint. And so the question is, okay, so we have this menu of options, and one could possibly address many of them. So what do we actually need to address? What are the OKRs in aging? So this, I guess, spoilers for some slides that I will be presenting in Toronto later this month. But if you, like, cause of death, we know what they are. It's going to be cancer, heart disease, I guess dementia, cardiopulmonary or pulmonary obstruction and things like that tends to also show up there for smokers, but kind of going down. And then you get into things like infectious disease for the elderly. That's another one happens, as well as accidents. Like if you're old and you fall, you may actually break a bone. Your wounds will heal slower and so on. And then you get into like a less defined list of reasons why being old is not great, like joint pain or arthritis or dementia or slower wound healing, or like your liver doesn't let you drink alcohol anymore because you get the hangover. You get all these things that if you ask even healthy old people, they will tell you, oh, this is not what it used to be when they was 20. And so, so one goes to, okay, well, how do we address this? Heart disease is one that I think we are not thinking much about. And I think it's one that there are already great companies working on this problem, because it's one that is very easy to address. It's very much like ApoB particles. It's all small cholesterol particles.
Some people make the argument that we actually already have a solution. It's just not being implemented in our healthcare system.
Yeah, I mean, I argue that like sufficient use of statins or very therapeutics,
inhibitors,
inhibitors, or even that in a way slows down sufficiently the disease. And Reason, who is a researcher in the aging field, is working on a company to engineer macrophages to remove cholesterol plaques, as well as a company that I think used to be called, or it's called Cyclarity, that they're using cyclodextrins to also extract cholesterol. Okay, so that's one way. I would say check, we got that. Then you get into infectious disease, brain aging, and cancer. Now we'll comes cancer to a later one. But then for infectious disease, we could say, okay, why is it that old people suffer more from this? I was actually writing on this morning, doing a deep dive. It might be that bone marrow transplantation, as in rejuvenation of HSCs, buys you enough to address that. Maybe it's all B cell aging and antigen-presenting cell aging, or maybe it's the lymph nodes. It might be that bone marrow transplantation with rejuvenation addresses that. And then you get cancer. Cancer is the and brain. Let's actually do brain first. Why is the brain aging? The brain is a low turnover tissue. You have these neurons sticking around for the origin of life. That one is going to be one of the hard mode aging causes. People usually don't necessarily die of let's say, unless you have Alzheimer's. So suppose you could address all these like other like Alzheimer's, Parkinson's. Suppose you have none of these. You still have like an old brain. What do you do there? So in this case, we don't yet know how to address this. There are things that could work, like in situ reprogramming for neurons. It's doable in principle. You can get things through the BBB, you can go into cells, you can do reprogramming. How well will this work? You could potentially do neural stem cell replacements, gradually replace neurons by adding neural stem cells and hope that those will then become neurons. A slow addition through your life of more young neurons. As the old ones die, you get more young ones.
Well, you've mentioned cell replacement a couple of times, but I think the one part of the roadmap that you haven't mentioned at all is potentially Nathan's favorite part, and also one that I think is undervalued, which would be the overall replacement. Nathan, are you with us?
Is it frozen?
Okay. We can hear you, we can't see you, but don't worry. I want you to push him here because that's an important area that hasn't been discussed yet.
Yeah, so replacing cells or bone marrow, I think that's one intermediate step that could buy us some more time. But if we're thinking about indefinite lifespan extension, and if we're thinking about, not just the cells, but also the extracellular matrix, then it makes sense for us to try and replace wholesale, like all of the tissue and all of the organs, let's say. And, evolution has already figured this out, right? Bodies know how to create themselves from embryos, and they're probably the most efficient way to create a young body. And that happens all the time, right? Sometimes it happens by accident, and it's de facto young tissues and bodies. So I'm curious how much you know or have an opinion about whole body replacement, if you could do some sort of cloning with some sort of neocortex knockout, and then do a head transplant, which some people are working on, I guess, and then we're exploring. And then, of course, that doesn't address the brain, but on the roadmap that we have for replacement, the strategy there would be to do some sort of progressive brain replacement with maybe like neural grafts, like engineered tissues, something that Jean Hebert is doing at Albert Einstein College of Medicine in his company, BE Therapeutics. So I'm curious what you think about that.
Yeah, so the replacement solution is one I find appealing, and it's also one where there is a gradient where you could replace cells, whole body, organs, or parts of organs. Cells are easier to start with, especially bone marrow, because the cells are so convenient. You can literally inject them into the blood, and then they will find their way into the marrow and then expand. Bone marrow transplantation has been a thing for a while, and this is possible also for other cell types. You can do it for intestinal stem cells as well. There are many of these cells you can replace, although this approach would require tissue turnover. So the new young cells can then replace the old cells, or you need some kind of injury to induce regeneration. On the other end, it is true that you have these more comprehensive solutions. It's like just get a young liver, get young tissue. I have not so much about I know it's in principle possible to do like this. You need one knockout where you got no neocortex or brain structures, and you get like a young body. Take a while for that body to grow. I don't know if you could accelerate that process somehow, and then you get into the brain issue as well. The thing with the whole body replacement would be that one, I haven't looked much into it to begin with. I don't have strong thoughts. It seems worth pursuing because it's really clean, and you don't need to worry about reaching all the cells and the ECM and so on. It does add the complexity of like the brain, the head transplantation problem. There was talk a few years ago of this guy called Sergio Canavero that was trying to do something like this, but it didn't happen in the end. I don't know if we have that technique perfected that well. I want to see more, I guess, animal work on head transplantation as impermanent, autologous or like isogenic, so there is not a rejection issue, and see if you can get them to live a long life. That will be, I guess, a first B0, just transplant, like old mice, young mouse transplant. That will also actually address the question of how much of aging is brain aging, because the brain is not just doing cognitive stuff. The brain is telling the immune system what to do, releasing hormones, other things. You could study brain rejuvenation if you do the swap, like young brain in old body or young body in a mouse, or like mouse with maybe surgery might be kind of gnarly, maybe in a rat or something. But yeah, so promising, worth doing. Maybe it will take longer. Suppose we begin working on this and what we're doing at Retro, what gets first to the clinic. I would argue that our stuff is more like it's closer to the clinic than this would be, and more FDA packaged friendly. I guess as an aside, roadmap of aging, there is the pragmatics of it, where you have enough money to solve the problem. You need to make some money to get the flywheel going. This is partly why Retro has like 5 bets, and then ideally, a single therapy can carry the entire company for a while. But first is like getting something approved. In our case, that's without going to the weeds of like not. It's like we want to have something approved that's not aging related. That would be just to make money. Then you have the brain problem. Jan Hevel proposed in his book Replacing Aging this idea of like you could cause a microstroke in the brain and then replace at some like some kind of like hydrogel scaffold thing and with some cells in there, and then you serially through these microstrokes you could like do it to the whole brain. I talked with Adam Marvelstone a while back, and I think we found like quite difficult, and in that so there is a lot of brain, and the brain is a 3-dimensional structure. So you might have to be in some kind of torpor state to pop up your skull and over many months or a year do this whole serial replacement. The risk for infections could also be high. You have to be in a very controlled environment for a long time, potentially. Because it's like the brain is huge, and in the paper I think is published, they were doing the area of each microstroke or something. It was like 1 square millimeter or something like that. So it's like you can imagine how long it would take to just the surface of the brain. It's just like the surface of the brain, the whole thing. I don't know how that has progressed since then. If it could be made to work, it could be nice, because people do get strokes and they survive and they are seemingly themselves. So you could imagine that you could keep this sense of quote unquote self while doing the ship of Theseus approach and slowly replace everything. Or you could do I don't know, it's like if this is one pitch, for example, and the other pitch would be some kind of crosslinks breakers plus neural stem cells and then gradual turnover of the brain with some growth factors. It's not obvious to me which one is best. I think both are worth pursuing. And maybe this is part of when doing roadmaps of aging. I've done roadmaps and bottlenecks work in the past with various people. One thing that comes up is that one could write a roadmap, but then one ends up with various interesting projects that one could pursue. And then it's hard to hand them to someone. It's like, okay, we'll work on this. People have to be true believers. Like, this is the one true approach, but it's not like you can prove it. People will get excited about different things. Ideally, the roadmap maker is also the one that takes a ticket from your Kanban board. Like, I'm going to work on the replacement part, or I'm going to work on the in situ part. And then you go and run with that for a few years. Both options are valid. There are no prior reasons why they shouldn't work. I think the cellular replacement or tissue replacement are closer to the clinic than the other approach might be. If it's coming to work, yes, it's a more comprehensive solution. It might be easier in other regards once it works.
Yeah, we exist in a socio-economic system which limits our decision making and affects our decision making quite a bit. what we're trying to do with our roadmap is focus purely on from a technical perspective, what would be the appropriate ratio of resource allocation, right? Because like when people ran the Apollo program, they weren't thinking about capital efficiency or anything like that. They were thinking about technically, how do we get this done the fastest?
Yeah.
I think both Nathan and I feel that the complexity of aging is still indeterminate, and the complexity of the solutions to aging are still indeterminate. And that's why it seems like it would be very important to put more resources towards both the replacement, the macro replacement, like whole body and brain replacement, or cryonics approach relative to the current distribution of the resources. I think the current distribution of resources is driven more by non-technical reasons than technical reasons. Would you agree with that?
I would agree that there is a lot of money going into reprogramming. Altos New Limit ourselves, we're already doing that. Who is doing full body replacement? And you could argue that who is pushing cryonics? You could argue that it's basically one small company called Lorentz, and not many others. Well, I guess there are a few others, Tomorrow Bioscience and so on. But yeah, I would say that the resource-wise, like if I had, for example, suppose I was a billionaire and I'm like, okay, I need to allocate money. I'm going to make one new longevity bet. What am I going to do? Person comes to me with I'm going to reprogram intestinal stem cells. Person B comes saying like, I'm going to do fundamental research to like body replacement, or like even engineer, even just making a livers. Like people, we cannot make artificial livers yet. I'm going to make, or I'm going to make artificial lungs, heart, muscle, or bones. I would fund those rather than the reprogramming one, because it's more out there and there is less money going. Maybe it's less FDA shaped, so to speak. Maybe it's more, the VCs will be more weirded out by the idea, because it's not a drug, it's not a device, it's a new tissue. VCs might be more conservative in bio than elsewhere, but I guess now maybe they're like alternative funding sources that will be more amenable to funding things like this. Like we had Sam Altman, for example, that funded us, or you had Yuri Milner funding Altos, or Brian Armstrong funding New Limit, so on. But yeah, on the like driven by social versus technical problems, I think that there's an old case to be made that if you want to do that the programming works and you can within reason get a lot from it, or like do it all the way without the need to do the full replacement. Especially for people that are already older, it's really hard to accelerate the aging or make a body grow very fast, to like accelerate the clock and make the body growing. Or I guess you could have a big head and a small body or something. I'd say, how young would have that artificial body have to be? Like 15 years, 20 years?
Yeah, you would want to start growing the body before you needed it.
Yeah, but then if you already need it like today, you're like 60 years old right now, what are you going to do? I think that approach makes sense. I mean, I'm not an expert, but maybe if you did the tissue replacement option instead, for example, you could say, well, I guess it gets really hard sometimes. Like, how do you replace blood vessels? It's not like you can take them out and put blood vessels back in. You need to get your body to make them for you, reprogram the blood vessels, reduce inflammation or whatnot. But yeah, I think that the approach is worth pursuing. I'm trying to think if someone that what hypothetically opposed this, what they would say. They would usually give ethical reasons, like this is weird, economic reasons, like the replacement solution is unusual, or scientific reasons, like there is no evidence this would work, sure, but like doesn't mean it cannot work. It's still like a parachute. There is no evidence parachute work. I don't think there are major arguments against doing it. I guess I can imagine reading articles against this, but I can imagine what they would say, and they would give bad reasons against.
Well, I hope a lot of people will listen to this, and I hope there will be some Altos and Retro level projects on replacement in the near future, because I personally would like to see that. So it's good. It's good to hear that you agree that it would be worth pursuing. I'd like to switch topics now and talk about the rate of progress. Number one is, do you see aging solved in your lifetime? And number two is, what are the ways we could accelerate the rate of progress? I mean, maybe you think it's actually going to be solved. I personally am pretty concerned. Start with that.
I usually don't do what I call timelines discourse. In SF, there's a similar question like, when are we getting AGI, artificial general intelligence? I usually am like, maybe, I don't know. Aging feels the same. I guess historically, if you were asked questions like this, when are we going to cure HIV, or when are we going to cure cancer?
Yeah. I've been in the industry for over 20 years, and we've gone from having 0 aging therapies that you can purchase to 0 aging therapies that you can purchase in that time frame, depending on how you define it. So rate of progress, I think, is something to be concerned of, although it's not like a linear thing, right? Like there's often step changes. And I think
Yeah.
those step changes often come with new tools. Advances in artificial intelligence are new tools, effectively. But there's other things like PCR, DNA sequencing, single cell RNA sequencing. Like we
Yeah.
tend to go through these step changes. I agree with you that timeline thinking
Yeah.
is difficult and problematic, but maybe you could comment on what do you think are the tools that we need that would make the
Yeah.
rate of progress.
It does feel slow as well. I read papers all day. And one often finds papers that, for example, or even things like the ITP from NIA. I do not think that that's that useful. For example, testing some molecules and it's like, yeah, we increase lifespan by 5%. I think that doesn't really do that much. And so it's more of a like longer term engineering problem that we have to say, okay, well, what? Instead of saying like, here's a random molecule or you screen something in worms, that's just not going to work. Probably some molecules are never going to work, almost a priori, which is so I guess to buy some more time or some more health, okay, maybe, but it's not like the thing. Similarly, I think that recently we have gotten a lot of tools to edit DNA, CRISPR base editing, prime editing, and so on. I don't think those will be key to aging in particular. Most people, when they are young, they're healthy. It's good that it exists because eventually we will need them to fix DNA mutations. But I don't think it's a genome problem either. I think it's probably not those tools. A key bottleneck, if one goes through the in vivo approach, is the delivery problem, the ever present. How do you get things to cells in vivo? But that aside, it's also just like more people working with the problem. For example, take the HSC one. Why are we working on HSC rejuvenation? Because you can do it ex vivo, you can put them in, you can get a big things like double or even 50% of all your cells are blood cells. That's many cells you can very simply make young. If only you could make autologous young HSCs. How many people are working on this problem? 3 labs, I guess us. That's it. That's mankind's thinking going into this problem. There are more AI startups than there are labs working on this problem. It's like, why are people working on this problem? And these labs don't have spin-outs to commercialize their research. I guess we're the one that's working on this commercially, as in it. Instead of being like, oh, it's an as curiosity, you can make these cells young. We're putting them in people to make them at scale and cheaply. It's like not many people are even trying to do what should be done. When I did one of these bottleneck analysis with one of the various iterations with Martin Borchensen, Laura Deming, and Alan Marlstone, we zeroed in on people working on the problem as a bottleneck. One could go to on the one hand, it sounds like a cope answer because one's brain wants to hear the technical bottlenecks, but on some dimension, people just work on their problem. It's like what actually moves the needle, both in discovering new problems and furthering work on the problems that we know are already there. Like, for example, how much do we know about replacement of the brain gradually through neural stem cells and growth factors? We don't. People haven't even tried. There are like 3 papers. We don't really know how feasible it might be. People aren't even trying.
The LBF did its own Bottlenecks to Progress survey last year, which we published recently. One of the major results from that is most people work on not what they think is the most important thing, but what they can raise money for.
Yeah, that's
or
another
get
one.
paid for.
Yeah,
Right. And like, so actually, like you could say people are the problem, but actually most of the people that responded to our survey were not working on what they thought was the most important thing.
Well,
The main reason was because they couldn't get paid to do the thing they wanted to do.
It's not like people are the problem. It's like people working on the thing, and then why are they not? One reason might be money, a reason might be interest or awareness. There's, for example, like you could solve some of that with community building, but you also need funding. You might want to work on aging, but maybe you don't have the money to do it. Community building is relatively cheaper than funding long-term research, and it will be nice. I guess what this goes into the should one go into like quant trading or something else, or do startups in tech, and then throw that money at longevity. It would be convenient if there were many billionaires throwing money at this problem, and there are more now than before, people with capital interested in this problem. The other interesting one, if you could get more of them involved, I like to point the example of the Whitehead Institute. It's this institute in Boston. This Whitehead guy, I think, let me see if it's true still. So no one or knows who this Whitehead guy is. If you go to the Wikipedia page, Whitehead doesn't have a Wikipedia page. Whitehead, the person after whom it's named, it's a random guy that happens to have a ton of money. It's not the philosopher Whitehead. So who are the other Whiteheads? Who are the other wealthy people that could be funding this? Random person in North Carolina that happens to be a billionaire for reasons, and maybe they could be convinced. I don't know. But yeah, funding is also definitely there. Funding coupled with the roadmap. Like if you had like here are these things you could be working on. People could say, okay, well, I want to work on this. Here's a bunch of funding to work on that. Impetus grants, for example, the premise of impetus grants, it's a pool system where researchers propose what to do and impetus funds them. But you should have, I think there should be push grants where the grant says, we're going to fund work. It's like, we know what you should work on. You should work on these things. If you want to work on the things that are part of the roadmap, we'll give you money, but not otherwise. We will not give you money to test rapamycin on dogs. We will not give you money to test this other thing because that's just not moving the needle. Maybe that's a bit of a hot take, but if I had grants, I would have opinionated grants. Doesn't mean being dictatorial. This means raising the status of helping scientists that want to work on things that, in the grantor's view, will actually move the needle. I think there are many people that want to work on these things, if only they had the money.
And I think that's what might be useful about this roadmap we produce is it could be a document that people can refer to be like, look, these are things that need funding. And then the funders can see that. And maybe some of those push entities could be created as a result of that.
Yeah.
Potentially by us even, but we'll see how it goes.
Yeah, I think that the framing that, for example, suppose one is extremely pessimistic or thinks that a limited lifespan extension is a pipe dream and it makes no sense, then if one has that mindset, one would say, well, what's second best we can do? And you could say, well, rapamycin or something simpler, and then you would find those things. But then, and then that like almost like difficults building this longer term roadmap, because sure, it's the road to sustaining increases in healthy lifespan is more unclear than tweaking a little bit here and now based on tons of existing research. Rapamycin is very well characterized as a drug, and there is replacement strategies that are less well characterized. And so science is often like, you want to work on the thing that you can show why it should work, or it is very clear, you can throw like 20 pages of evidence why it's going to work. And so there might be some slight conservative bias in general. I don't know what the sociology of like, why are we not being more wild than academia is. I guess that's the one. Like academia is, due to incentives, slightly more conservative, and then companies usually don't do long-term fundamental research because VCs won't fund that. They want something that will get to the market. So then we rely on academia for the wild stuff, but then academics don't get that much funding. So we might need a crazy labs set up for aging that funds crazy stuff. Now you could say, well, we have things like the Back Institute, that's an pure aging institute, but they have produced lots of great work. But if they are not driven in this way, they are not like, here's our roadmap to solve the problem. They're exploring things here and there. Why? Well, I guess it's partly, if you look at how labs are structured, even if you want an extremely wild PI, you still need your students to have papers such that then they can go in industry or academia, and then you cannot impose risk on them. You need an FRO for aging, or you need an institute that's like as opinionated. It's not like a bunch of labs. It's like it's Manhattan Project S, or I would say not Manhattan Project S. In a Manhattan Project or Apollo program, the goal and the how to get to the goal were kind of defined-ish. It was much clearer how like ABCD, here's the plan, whereas this is going to require some more exploration. So it's something that might be more like Bell Labs or Bell Labs.
Yeah, I think the Weiss Institute in Harvard is a good example. That was also mostly funded by, I think, one basically rich person, and has produced like a large amount of the synthetic biology tools that we now take for granted through George Church's labs and all the other labs.
Yeah. So Jose, you mentioned we need something more like a Bell Labs for aging, but isn't that kind of like the thesis of Calico and Altos Labs, that they were trying to be Bell Labs of aging?
Well, I mean, one is doing, so I guess Bell Labs, what makes, because you could say it's Retro also like one. So what makes a Bell Labs? So you had AT&T, they have like a series of like product lines, and then they have researchers that are researching things. And they can do that because in a bluish skies way, but within the constraints-ish of like, your thing eventually has to make it into a product, ideally. If you have enough products, then most random research in that direction probably will end up in one of the product lines. If you only have one product line, then you cannot be as wide in your research directions. But that's what should be there. Since there should not be students at this such place. Everyone is it's a member of technical staff and an engineer, and then there are like roadmaps and goals, and like there's an end goal in mind. We want to solve this problem, and here are 3 approaches. You could argue that AI labs like Anthropic or OpenAI are closer in that, and there's a goal which is like safe AGI. It's like, okay, cool. How do you build AGI? Well, here's like, I guess OpenAI is more of the try many things at once. Anthropic has more of a defined perspective, but they're like coupling product development with research. They don't have it's like, here's exactly how you make AGI. It's like, here's paths where you might get there, and then you explore them, and then you sell things along the way. So and these companies like can afford to hire really good talent and put money back into their operations by selling some products and having some revenue. You could imagine, though, suppose that Warren Buffett is like, you know what, longevity is super important. Here's $100 billion. There will be no need for a product. You could just go and do it. But that's the thing. How likely is that?
Do you think $100 billion would be enough to get us?
Yeah,
or not?
it's a lot of money. It's a lot of money. Like Retro, for example, is like seeing how much we have achieved. We have the funding, which is all that's publicly known, and you can do a lot with that money. It's like once you get to the clinical trials on the road, sure, things get a bit pricier, but up until then, you can do a lot. And even if you pay good salaries to scientists, once you have the initial one-off capital cost setup of your lab, or if you are like clever about it, it's not that expensive. I think it's much cheaper than training huge AI models. It's more like finding the people willing to work on this, and just like, again, for example, for us, for our HSC rejuvenation program, want to hire people that have the knowledge to work on this and want to work on rejuvenation. How many people are there like that? It's not like there are a million. It's not like hiring, you post a software engineer for our applied AI team, and a million people have GitHub experience working with PyTorch. It's more, with HSC it's more difficult. And you're also to some extent buying tacit knowledge from them. This person was at this lab, they know how to do this thing, encounter all these problems and know how to debug these problems. And you need those vague things to make your program work. So I would say hiring is hard. Like you can hire everyone you want, and then you run out of people to hire to some extent. And then you get into building automation. That's the next part. Automation is another constraint. Like you can actually throw money at that. You could say, well, you have cell type A, cell type B. How do you make A into B? Well, we could try these 5 variables. Cool. Nice. We have a robot to test them all in combination now. So that's, yeah, building an automation core takes a bunch of money too, but 100 billion, it's a lot of money. $100 billion, like it's so much. It's like you could even buy your own buildings, you could buy your own sequencers. Even if you spend like, it's like 1 billion a year is a lot of money. Like the thing with bio stuff, as a magnitude, Blue Origin, I think, was spending, the rocket company from Jeff Bezos was spending that a year, and there was like hundreds of people, and they were like buying very material heavy stuff. Biology materials are not as expensive. The amounts you require are tiny. If you need to make AB, it just makes itself. The concern with bio is just being able to try more ideas and knowing what to try, and knowing when to give up and not to give up. Like, for example, should you're like, we're going to try to add this cytokine to this cell type to make it do X. It's not working until someone comes and says, but wait, the ones from this company actually suck. You have to buy these other companies. Or like, it works, but only when you combine it with these other things. How are you supposed to know that? So that knowledge is key. But yeah, $100, you can have the right people, right equipment, and you can probably solve the problem, I think. Or at least like go to a point where you could get so close, you could just fundraise all you want after that.
So you mentioned 2 things that I want to follow up on there. One is the types of people that are hard to find, because obviously one of the things the LBF is doing is trying to bring more people into this industry. And so I'm kind of curious as to what types of people are the hardest to find, because we can specifically try to seek those out and bring them in. And then the second thing is about the computational and automation side of things. I mean, you guys have Ben Ray there, which I know he has some pretty ambitious ideas.
Yep.
And I do think that like there are certain circumstances right now where we've created closed loop, fully automated software and hardware driven processes, like trying to map all the ways to turn one cell into another cell, right? Like you can solve that problem by adding more machinery, adding more compute. It doesn't necessarily require more people. So yeah, 2 questions. What are the types of people we need more of? And what are the types of problems where we could, in the foreseeable future, start not needing people, where we could create these closed loop situations?
I think on the people, I would say if you break Retro, for example, there's the operation side, which is things like facilities, keeping things working, fixing instruments, receiving, shipping, scientists doing the experiments, surgery and the mice, keep track of them. Then you have computational biology and maybe software and automation. I would say the scientists are the hardest to find, and that's, I think, because the barriers to entry. I guess it requires a specific kind of person to be willing to put through the grind of like bench work and be okay with doing that. I guess the average software engineer will be like, what? You have to be in person? You cannot work remote? You earn half of what you make at like Google? Why would I do this? Fair enough. Well, because there's no other way and you have to do it. Or maybe you find it relaxing. I don't know. But it requires a specific kind of person that's both very smart and willing to go through that grind in a way that's hard to ramp up yourself. Like if you're at home, you can become an okay software engineer. There's so many resources. You can read code base, get remote programming, many things. But lab work is quite hard to pick up on your own. Even though you could teach yourself how to do PCR from home potentially, and then pipette, and yeah, sure, do it. But the tacit knowledge of having seen things when you do a complex experiment, that's much harder. There is not much. It's not like people could GoPro themselves while they do experiments, and you could watch tons of videos, but we don't have that. So I think scientists are the hardest ones to hire. This is, I guess, in general, in biotech, I don't know if aging is worse or better in that regard. I think aging might be easier because aging has the mission aspect to it, the big quest of like, we're an aging company, as opposed to something like making this tool, making CRISPR a bit better. Maybe it's less inspiring. Some people that come to Retro, they really want to work on aging. They're very passionate about aging. They might even take pay cuts to work on aging. I used to work in tech, and the pay is different, but the meaning is higher here. So on the automation end, our automation team this last year and this year, the thing with automation is that it can be very expensive. If you don't know, if you could like, as some companies have done, blow $100 million in automation and then be like, oh shit, it doesn't work for us, or it's not what we wanted. And so we have instead, by us, is Ben who has been leading this, figuring out, okay, what do we actually need? It's like, what's the cheapest you can do? Can you use an OpenTrans? Is it good enough? No, it turns out. Is Hamilton's good enough? Can we like using like, like our own parts and like attach things together and write some custom scripts? Can we make work cells that can do generic work instead of having to reconfigure things all the time? Once that's working, you can say, okay, well, now we have this work cell. Now we need to multiply it by 5 and build software to actually use them in some way. We'll probably be hiring more software and computational folks this year. One of the bottlenecks to lab automation is that both the hardware is cursed and the software is cursed. Like some of this, let's say, for example, the V-spin, which is like automated centrifuge. There are not many automated centrifuges in the market. You have to best buy that one, and then you have to deal with like Windows random DLLs. It all runs from like Windows computers you have attached to these things, and then the drivers, you have to write code, I think, sometimes in .NET or stuff like that.
Very familiar with that specific problem. Synthago built its own automated centrifuge because we just couldn't find one that was.
Yeah,
it was easier to design and build our own.
It's so cursed. I mean, maybe I shouldn't suggest that because we will actually do it. but or like if you want to, for example, do something as simple as tell the centrifuge open door. Ideally, you want to query, is the door open? If so, then put sample. But some things you cannot do that query. You have to wait and assume it's open. Most times it will, but that sucks. It's that you should be able to query the state and not allow illegal states, but you can't do that. So yeah, you either galaxy brain your way through computer vision to see like it's actually open, or you build your own. It really delays people because then you have like, you guys build that thing. Now we may have to build it ourselves again. It really sucks having to do all of these things. Plus, on the software side, the standard for coding and automation is usually not great. It's usually built for one workflow. For example, you may have 8 samples and then insert a for loop A1 to A1 on this plate and so on. But if you have 7 samples or 9 samples in a different row, you're fucked because you have hard-coded all the motions. It's like writing code by using pointers and never using a heap allocation. It's insane. What we might do instead is write a compiler that compiles the protocol in the abstract for any number of samples and it allocates where things should move. The user should not think about plates or holes. You should say, I have n samples and I need to add this much to the samples. And you should not think about where things are. The program will tell you where to put things and it will figure out the inner routing itself. That's how it should be. That's how it's in software. And this is doable.
Yeah, it is in the tech industry. And I think the tech industry, they have a lot of money and they subsidize the development of open source software and standards and things like that. Yeah. And that's not really the case in biotech. I guess, is Retro interested in like sharing? Because like it's like every company seems to have to reinvent its own wheel.
Yeah.
And like that seems kind of frustrating and it seems like a bottleneck to progress.
Yeah, I mean, maybe we have open sourced some things in the past. We have open sourced our OpenTrans 10x prep robot that Ben released, I think, last year.
Yeah.
We might do more. I think it's good for recruiting. And it's not something that gives another company a huge advantage. Well, I think we also have some contributions to Pilot Robot and by Hamilton that we have made, and because we just needed to improve those packages to just make them work for us. But I think with automation is that many companies, or maybe do not believe in automation. They just do not even try. They think it's either too expensive or too rigid. They're not willing to go hard on automation. So if they are not, our package will not really help that much. And amongst our competitors, I don't think that others are thinking as much. about it. It depends. I don't know if we will open source more than we have so far. We might, especially this like allocator I described. We might also open source other tools that we have built for computational analysis of stuff. People use things like ScanPy or Surat in R. We have built our own that integrates many methods into one. When people do differential gene expressions between cell types, and it's like, how are these cells or cell clusters different? There are many ways to do that. People just usually use one. But if you run 7 methods in parallel, they will sometimes give you very different results. And it's like, what's real anymore? So we can run them all in one command without having to worry about their parameters. Just like run them all, score them, what's consistent. Not ideal, but it's something. And we have built tools for stuff like that. Also working on perturbation prediction, given initial cell state, given desired state, what do from cell A to cell B. But that has to be also done in a broader context. Like it's one thing to say, like, suppose you set up a screen and the order model tells you factors are useful for rejuvenation. It's like, cool. But then you have the question of how much of the factors, when to add them, what media to culture the cells in. Maybe you need to have media for iPSC between at day 3, once you are pushing the cells towards the programming, because if not, the cells die, and your model doesn't know about media. Whereas if you have automation, instead of having a computational approach, it's very tempting to narrow your problem so much that it becomes not realistic. The classic example is you have a protein, you mold the protein, and you throw computation at molding the protein at what binds or abstracts a kinase, what pockets. But then the question is that kinase actually what you want to hit? How much are you solving there? And so on. Or the extremist test in vivo, testing a bunch of mice. And in the automation case, if you're doing cell therapies, you can actually iterate over what media, how many days, what cytokines, what factors. Computational models can give you lists of factors to screen in combination or alone as part of this whole experimental optimization with the culture conditions and a couple of other things. And I think this is only really possible with automation. I think computationally it will be quite difficult because you would need data, and to need data you would need automation to generate all these different conditions to begin with. So I think automation is going to be key, and we will probably see more. I think not from only us, but from other companies. The latest OpenTrons, the OpenTrons Flex, looks interesting. And there are a couple of other companies like Medra or Trilo Bio that are also pushing on lab automation. And I think it's like maybe there's like a cultural from the bio side. It's like robots are hard to program. You need someone to work with them, and then as long as you need an instrument that doesn't fit in the deck of the robot, we have to fall back to manual. So there are many problems to solve, but none fully addressable.
There's a bit of a culture gap between the software, hardware people, the biologists that I think is starting to be closed at certain companies. Certainly Retro is an example of that, but it still is predominantly like they're sort of separate groups of people, which is a problem. But you brought up an interesting, which I wanted to get to while we still have time, which is models. So another example at Synthago is we had a model where we were doing random perturbations. Every time we ran a synthesis, we would change small things like temperature, pressure, concentration, time, and we would do basically perturbation studies on our whole process and figure out, like, optimize the process both for cost and quality, and managed to make some like incredible improvements over, what humans had discovered through human-based experimentation. That works really well when you have, that's a narrow problem. This idea of building models of biology and then training them using perturbation studies, is that something that you guys are actually doing at Retro? And then also, what types of computational models? Because obviously the transformer LLM type thing is very popular right now. I really enjoyed Lex Fridman's podcast interview of Stephen Wolfram, because he was talking about how the language model is often what you do when you actually don't understand a problem at all, and you want to eventually switch over to more of a symbolic logic thing, because that's more computationally efficient and it's mechanistic. It's like you actually have figured out what the mechanisms are. So like there's a lot of different types of models. And what do you see as the future of modeling in biology and moving more work into the dry lab versus the wet lab?
Yeah, I would say that like on that, it depends, but sure, if you have a symbolic model, it is better in those ways. But biology is a hot mess. In principle, you could have a series of stochastic differential equations modeling the ways in which everything bumps into a cell and splicing.
Biology. I think we're going to make a T-shirt out of that one. Actually,
Yeah,
you want that one?
it's really cursed. I mean, there is like a piece of art I have here that, like, it's just like, it's a bunch of squares that look very weird, but it's the optimal way to fit the squares in one big square. And it's like, oh, this is like, it represents that, like, sometimes the way things are not the way you wish they were. They're like fucked up, and that's what there is. It's like, you have to deal with it. I cannot go into details about what you are computationally doing at the Retro. I will say that transformers are very nice because they are very efficient. You can just shovel data at them, and they will take the data in, and they scale very well. This is the thing. You could, in principle, train other things, and with enough compute, they might be as good as a transformer, but it just might be too expensive that it might not. Now, it's depending on what you are doing. Like, some people do, like, let's say, do like sequence work. You have like proteins, and the transformer is attending to like those sequences. If you're doing gene expression or chromatin, it's different. In that case, you don't have a sequence, another sequence. You have like a bag of mRNA, so you have like a set in like an invariant, like position invariant transformer, or you have your this is, I guess, compared to like the language ones. You have, for example, a smaller vocabulary, like there are only so many genes, for example. So your vocabulary is always like, let's say, 60,000, and your context window is always 6,000 because it's always like those genes. So these models might look different from the ones that one would use with text. But yes, there are many papers that have come out this year and last year now. There's SGPT, there is GeneFormer, a couple of others, using techniques similar to what has been shown to work with text. Things like pre-training, for example. You can just say, okay, here's a cell, just mask a couple of genes, try to predict those genes from the other genes in the cell, and you pre-train that way, and then you can tell the model, okay, here's a cell, and I'm going to artificially increase, let's say, OSKM. I'm going to set those values in the input to like very high. You give them to the model. What does a model output? Tends to output a cell state that looks closer to an iPSC. So the model is kind of saying, like, this perturbations seems to push towards iPSC. So you could run a screen over your input and then see what's pushing the cell more. So this is without having any perturbation data. This is a perturbation data set would be input output cells plus perturbation. So it's a triplet. Whereas in the pre-training case, you just have like just like data. It's like a matrix of like genes and cells, which now last year CZI released this big corpus called Census. It's like 40 million cells of all sorts, and people have been training on that. But there's not much data in general, so you have to produce your own data, and the data quality is also not great. So choosing what data to generate, how to generate it, it's also important. But yeah, but be that as it may, so this kind of work can get you lists of, you could say, on the one hand, you can produce lists of targets from this. You can say, oh, these factors seem useful. But you can also just do a screen and just try them all. It's like, there are many genes to try, but you could say, well, if you want to rejuvenate a cell, is this one random gene actually going to do that? Because it might be that if you apply a small amount of thinking about your universe of things you could try, you can actually do many of those combinations directly in a dish. And so the model might help you pick a list of, like, doesn't have to be perfect. The model can tell you, give me the top 1,000. And then it's like, cool, now it can test them all. Or like, and then you can pick your top 5 hits and then do the combinations for the top 5 hits, titers. So yeah, but we are far from the Bayesian, like 24/7 hyperparameter optimization over every possible thing in an experiment. But that's, I think, to me, the goal. Once you have automation running 24/7 with like all the tiny problems, there are like so many tiny problems to solve before you can get that working. Like a random example, it's one thing to load an experiment and having run for a day or two where you have everything you need and you can just go home. But if you have things like media changes and you need to keep, or adding antibodies at day 4, you need to keep those things either in a freezer or in a fridge, and then know where to take. The robot has to, if you want full automation, it has to be an automated freezer, an automated fridge, and you want to thaw at the right time, and you need to aliquot things beforehand, or you need to like uncap or cap your. There are so many like tiny things that need to be figured out, but they're all known. You try to experiment, what doesn't work. Okay, how do we build this? And you just like try. It's always fun talking to a scientist to do like a process mapping of like if you take flow cytometry, what are the inputs, what are the outputs, and you get a map, and then you're like, well, sometimes you do everything, but it's like, and you keep adding until they're like, yeah, this is basically it. But it's a fun back and forth because at first they know it so well, they don't have a formal map in their head. It's almost like a really good chef that just cooks very well, but they cannot explain exactly what's going on.
Yeah, this is what I call the difference between sort of like the traditional way of doing science. I call artisanal science.
Yeah,
actually.
yeah.
And because you don't even, yeah, like if you asked them how they did it, they would give you a list, but it would be like only 20% of what actually matters, because so much of it is
Yeah.
not encoded.
Yeah. I guess it's like on the computational side, I guess we might see some more, because New Limit, for example, they're going, I think, from our view, much harder on they're like computational first, or computationally driven, than as far as computation is one thing among others than we are. So I guess if we see if there are like huge gains to be made in T-cell reprogramming, or I guess now they are also going towards HSC reprogramming or and liver, we might see it from them sooner. Whereas, yeah, because I think if I understand correctly from Blake on Twitter, they want to find and sell factors to others, find combinations and then sell those. Whereas we want to like sell therapy. I want to like say, here's the retro cell, and you put the retro cells in you, as opposed to here are the retro factors, and then you put the factors in cells, and then someone else like someone has to make a therapy at the end of the day, and we want to make them.
All right. I think that covered a lot of the computational stuff. Nathan, did you have any other? I know cryo is one of your favorite areas as well. So
yeah, we can discuss cryo a little bit. You kind of mentioned that people should be working on it, and there's only really like one or two companies really working on cryo slash cryostasis, let's say. But for some people, their mental model really is just like, hey, biology is just so complex. I don't think we're going to make that much headway on the problem within my lifetime. And they're just going to do what they can in like maybe 10, 20 years today, and then switch to cryo, basically. And that's their mental model of like how they want to attack this problem. But, cryo itself, some people would push against cryo and say, well, this seems also kind of like a pipe dream, and, it hasn't been proven out. It's also theoretical in some sense. And maybe you should just work more on, let's say, like traditional rejuvenation, biotech, because that's a little bit more tractable. I'm curious what you think about this. Like, you think success is a theoretical possibility?
Yeah, I will preface by saying that I don't have deep knowledge of cryo in general, so my opinions have to be discounted accordingly, and I cannot go into the weeds of either the field or what's promising or not. With that said, I guess there is the observation that some have made that some animals freeze, or they cool down over winter and then come back, like some frogs. You have, of course, tardigrades and other smaller ones. And we also know some things about cryoprotectants like trehalose or some antifreeze proteins that some animals secrete. We also know kind of the like, you could say, well, if you put a human in ice, what's wrong? We know what's wrong. We know what happens. We know like cell membranes burst from the ice. So we know kind of the things that we have to be watching for. Kind of the what to do is, or rather, what to fix is kind of known. How to fix it is still unclear. And
but there's clear benchmarks, so it's more of an engineering problem in that sense.
Yeah, it's like, and there has been progress. Like we have been able to, let's say, freeze. I don't know if it's like a human liver or a rabbit liver, but like big like at least organs that we have been able to cryoprotect and recover. Greg Fahy, who is, I guess, known in some areas for his work on the thymus, is also a big, or maybe most known before that, for cryopreservation. Is in, I think, periodic prizes given to him for his work in the area. But yeah, I think, like, for example, okay, well, take Alcor, for example. Alcor has been around for a long time. Should someone sign up for that? I mean, Laura Deming's words, maybe not, because maybe just like, although, you could say, on the one hand, sure, Alcor doesn't have a plan for how to unfreeze you. They're relying on like, well, maybe we'll figure it out later, and then so you're making that gamble. Whereas I think maybe one could make a case saying like, well, maybe it's so destructive that the technology to undo that damage is going to be so out there that maybe just like cannot be done. So can we find a way to more gently freeze things? Ideally, show in our lifetimes you can freeze and unfreeze someone without damage. I don't know what Lorenz Bio, which is Laura's company, I don't know what exactly they're up to right now. I know they were like trying to replicate to establish some knowledge base from what has been done before. But also, I think that in the cryo field, there seems to be two lines of thought. One is the classic, like you get frozen and then you get unfrozen and then you go on with your life as you were. And the other one is what they call the fix and slice approach. This is where you would not freeze, but like fix in formaldehyde the brain, then slice it. and then scan it and upload it. So it's something that kind of has been maybe an undercurrent in that whole area. It seems like giving up about the bringing back your embodied part. It's like, well, this is too hard, but at least we can store the information in your brain maybe. Then gets into philosophical debates about like, is that you, or like, is it worth doing that, and things like that. And then but on the other hand, it seems more doable. I think scanning a brain, once it's fixed, it's reading it seems easier than unfreezing it in the right way and bringing someone back alive and so on. But yeah, I don't have really, I guess, broad thoughts on that. Other than, yeah, I guess you could say, well, if you ask me, it's like, okay, well, how would you make it work? I would say there is the perfusing with right cryoprotectants seems to make sense. Using mRNA and very deliberate methods to get all of your cells to produce cryoprotectant and like have it be inside all of the cells also makes sense. It's probably don't need to cryoprotect every single cell as long as most is protected. Even if some cells die, well, that's fine. It doesn't have to be perfect, because maybe by then you can maybe replace those. And that's pretty much.
Yeah,
And I guess also maybe from the. So I remember talking to some people about this a while back. There's the issue of rewarming someone evenly. You could maybe address it by, when you're perfusing with your cryoprotectants, you could also infuse some kind of non-toxic metallic spheres so that you can use microwaves.
you can use a form of radiation that can go through the entire body so that you're actually heating everywhere rather than just like from the outside in.
Yeah,
and that is actually how they did that rabbit liver
the rodent technique, the nano warming technique. Yeah, used iron oxide.
Yeah, nano warming is the key word there. Yeah.
Magnetic alternating magnetic fields. Yeah, I guess it's interesting though, because like, yeah, you brought up an interesting point where it's just like, yeah, what are the things we most need to cryopreserve, right? Because some of the cells, we probably could replace, maybe even the whole body could be replaced, right? So it all comes down to the brain again. And, I feel like a lot of these roadmaps, the critical bottleneck is always, or the last frontier, the hardest problem is always what to do with the brain, right? Whether we need to rejuvenate it, replace it, or cryopreserve it, it's always the brain. So
the brain.
Yeah. Mark, are you?
Yeah, so I think we've covered all of the major categories at this point of the roadmap. I guess, well, one more before we get into the sort of final thoughts section is like maybe modalities. So a lot of the industry is obviously focused on small molecules, and I think a lot of that is actually due to, it's a historical artifact because there was a lot of breakthroughs and successes with small molecules in the 20th century that solved 20th century health problems, but it's not necessarily the right tool for the job for something as complex as aging. Like, for instance, even with reprogramming, I've talked to people in reprogramming, and obviously like the Ocampo lab has put out some results recently. And when you do this sort of just across the board reprogramming, that's off probably a net negative, right? And so like ideally you would want a cell by cell level control.
That was also continuous reprogramming. So that paper was the point to like push it to the end and see how it breaks.
Yeah, but ideally you would want
you're right.
A gene delivery type thing where you're delivering something that can perform logic. It can tell what type of cell it's in. It can tell how much of a nudge is appropriate or optimal. And then you run into the gene delivery problem.
Yeah.
And so to me, that's one of the biggest obstacles. If you're going to go not with cryo or replacement, doing large payloads without off-target modification, like large complex payloads and getting to good biodistribution is probably one of the biggest problems that we have. What do you guys, do you have thoughts on how we can solve that problem?
Yeah, I guess there are like 2 or 3 mini questions there. One is the modalities as molecules or things. Then there is the reprogramming, in particular uncontrolled reprogramming, and there is delivery of cell and gene therapy. On molecules, I think they are maybe easier to synthesize, or historically they were easier to make at scale and cheaply. They have been very useful as well. They have done so many things for us, surprisingly to some extent. One model of how they work there basically is like taking one lever and then it's like do a bit more, like a bit less inflammation, a bit more of this, or like make a bit less cholesterol. It's like one thing at a time usually, and in a very acute context. You have some more chronic things like statins that are supposed to be taken lifelong, but almost by their nature, they are basically when you have a drug, the drug has to basically have selectivity for the thing you want to hit, and then efficacy. And then in other cases, the additional complexity of bioavailability has to be orally available as another constraint. You need to encode this information in only so many atoms. It's very small. Whereas with a protein, for example, you have like so much to work with. You can make a protein that acts in a specific situation because of its shape and does everything you want it to do. With a cell, you even have more degrees of freedom. With a CAR T cell, for example, you can say, kill only if you find this antigen, otherwise just be chilling. You cannot, with chemotherapy, you just cannot do that. You can hope that cancer cells replicate a bit more, and this is why the chemo affects them more, but it's not as precise. And you have, of course, CRISPR, you have like a literal ticker tape of a guide RNA to say edit here and only here and don't touch anything else. So I think the future of medicine is like molecules will eventually fade away to some extent, modulo some constraints about cost and price of these other modalities. Ultimately, a lot of medicine is, or like if we solve, for example, metabolic disease with things like Ozempic, that like get rid of a lot of use cases for molecules. The reason people get sick is partly metabolically driven. Suppose a world without diabetes, or a where you're just healthy overall, and of course a world where you're younger in general. So I think for aging, the therapies that people will end up using will be like either weird ones like replacements, like the therapies like you get a new organ, or the therapies as gene therapy, or rather an epigenetic therapy or cell therapy of some kind. Well, one could say, sure, but there is chemical reprogramming in vivo, as you cannot control the chemical reprogramming. It's just molecules just doing what they do. So you run into this problem, it's the control problem. If you're doing a therapy where you want to reprogram in vivo, you need to make sure that cell by cell, you're not pushing it beyond where you want to push it. At Retros, we have an in vivo reprogramming program, and that's very key to us. This logic, this cell by cell control. So it's not just deliver Yamanaka factors, switch them on, redox cycling, and then that's like, that's it. You need to make sure that it switches off when it's gone too far. There are many, I guess, genes you can be looking at for like what too far means, and you can like, I guess, define various. You can have like multiple like switches for like, okay, this is like, or even you could have like a late stage. Okay, if you have nano activation, just like kill the cell. There are various ways to do that, but you can only do this with genes, with nucleic acids. The logic you can put in a molecule is just like not that much, just by just like they don't carry that much information in their structure. I think the thing with bio, you have to encode the logic for when it acts and the action itself in a physical thing, which is not the way we're used to thinking about stuff. CRISPR is nice because you can decouple those. You have the same Cas9, and you have like you can exchange the what you're editing with the guide RNA. Back then, you had meganucleases where you were encoding the editing in the structure of the thing, which is why they were so hard to make. So yeah, I think that's a future for aging therapies. It's going to be nucleic acid based, broadly construed, either things done to cells that you put them in, or things you do to the body with therapies. And then I go back to I guess like many of the things will probably have the shape of like either reprogramming or enzyme replacement. For specific things, they might still use molecules. Like it could be that to remove atherosclerotic plaque from the arteries, modified cyclodextrin as a one-off every few years, that's the job. Or it might be that to break a specific kind of crosslink, as molecule or a kind of protein replacement where you inject a specific protein or enzyme, that could work for specific things. So I'm not like it has to be one or the other, but yeah, given what aging is, I think at least at cellular aging, I think for cellular aging, you cannot hope to fix it with molecules. I think it has to be either an entire replacement or something like reprogramming, where you need like multiple factors, or ex vivo reprogramming.
Very cool. Yeah. I mean, I agree with that. There is a list of targets that you can imagine, and some of them are addressable by small molecules, but probably that you'll hit a limit pretty quickly if that's your only modality. And so we do need to do that tool development. So I think it's time to get into I have a list of final thoughts, unless Nathan, was there any other roadmap aspects that we missed?
There's one thing I wanted to ask you about. We've been talking about goals and different pathways on the roadmap, and we discussed the current state of the field. One of the things I really like about Retro is how Joe frames the mission of the company, which is very distinct, very measurable. The goal of Retro is to extend healthy lifespan by 10 years, and then you can ask, okay, but whose lifespan are we extending and so forth? But I'm always curious about this, like people working in the field, especially when they're thinking about disease indication, but then also the greater goal of actually extending human lifespan, like actual longevity. How do you know that you've succeeded, like to extend lifespan by 10 years? How do we get that readout? Is it going to require aging biomarkers or lifespan trials, or is it going to be epidemiological data, just like, after the fact, people are just taking these? I'm curious what you think.
Yeah. It's like indeed, there's this document I wrote internally about the mission of the company. How do we know? And it's like there's also a question for whom. I guess informally, the for whom is someone that's healthy, someone that has done everything within reason to stay healthy. If you are lean and are exercising and eating healthy and so on, you will still get Alzheimer's maybe and cancer. You get those things. So for those people, it's the health of those people I want to extend the life of. For everyone else, it's Ozempic, I think. So we want to work on the hard problems, not the easy problems. It's harder to make a healthy person healthier than a sick person healthy. The gold standard trial eventually is basically hypothetical. I'm not saying that we will actually run this trial, but if you want to see it for sure, you literally take a control and a treatment population and then test. You have the retro therapies and then non-retro therapies, and you just like do the lifespan trial, and then you see that on average lifespan curves show a separation of 10 years. That of course it will take a while to run. So instead, if you take the current retro therapies, for example, if you take more pragmatically, our T cell team is working on a specific approach to address cancer by rejuvenating T cells. T cells are made when you are like up until, I guess it's like 30 years of age, then the thymus stops working. You have the T cells you had when you were 30. They are the same T cells that will be fighting cancer 30, 40 years later. It's those same cells floating around. I want to make them younger. So in that case, you could say, well, is cancer a disease that a person that's a healthy elderly would get? Yes. If we cure someone of cancer, will that person live potentially 10 years longer? Potentially. So in that case, it could count. But you could say, well, that's a very acute disease. It's like it's dead or it's alive. Cancer trials are very friendly from a regulatory point of view because they will let you try more aggressive therapies in patients as a maybe third, fourth line of treatment, and then it's literally, if it doesn't work, they're going to die anyways. It's very acute. But then you have our HSC program. How do we show it's working? Heuristically, the cells are younger, and they behave younger by various metrics. In mice, they do extend lifespan. These younger HSCs. It's like, sure, if we could translate the percentage of lifespan improvement in mice to humans, then you might get 10 years. But like, we don't know how well it translates to that. We could say, well, we know in humans that get bone marrow transplants, you randomly get, sometimes you get young, sometimes you get old because of the way transplants work. I mean, patients that get a young transplant live longer, somewhat longer. Will a healthy patient that gets a transplant live as long as the patients that got the transplants in the condition of cancer that we have seen in the population? We don't know. We can only hope that given the mice data in wild types and given the human data, it will work. We can also do interim things. For example, if you have a young immune system, it should be the case that if you get an infectious challenge, you will produce antibodies faster. There are things you can measure in the shorter term. And then if we do that test, then we can expect that your mortality from infectious disease will go down because we have fixed that one. We have like in that very narrow way. So we basically, we cannot hope to do a longevity trial to show that. We might be mission accomplished way before we do a longevity trial based on reasonable evidence. I think based on the causes of death or like causes of like, because it's 10 years of healthy lifespan. So if you, for example, if you have arthritis and we reverse arthritis by some means, well, you will feel that you don't have arthritis anymore. So that's like, and then you can track how long are you arthritis free, and is it 10 years free of arthritis? Then that could count. Because ultimately, if you're extending someone's life, you have to be addressing heart disease, cancer, and these sort of things. What are biomarkers for cancer incidence? How do you know that you have reduced someone's cancer incidence or you have increased their odds that if they get cancer, you will actually fight it more effectively? It's kind of unclear. If someone is metabolically healthier, they seem to have lower risk of cancer incidence, but they're really assuming that their patients are metabolically healthy. I guess there are, for example, DNA mutations. You could think you are going to get more cancer if there are mutations in genes that are oncogenic, but we're not addressing DNA mutations, so we cannot use those biomarkers. The only thing we can do is to develop therapies that if and when cancer occurs, we're going to let cancer happen and then we will address it. Can we slow down the occurrence of cancer? Maybe. You could potentially. Rapamycin, for example, it slows down protein synthesis through the mTOR. But if you are on the reversing aging point of view, and reversing means treating cancer when it happens, you could combine prevention with treatment. In that case, the trial becomes easy. Is there cancer or is there not when you do the trial? So in that case, you have to, and I think for cancer, that's the best approach. You should assume you will always get cancer, then your trial is easier. Well, you get cancer, then is it cured or not? So the question is, how do you cure cancer? With the cancer case as well, it's an interesting one. Why is it that young people don't get cancer or get cancer less? You have childhood leukemias. Is it because their immune system is stronger, or is it because they don't have as many DNA mutations? I tend to think that it's mostly the mutation story. The immune system helps contain cancer, but I think even if you could fully rejuvenate the immune system, as we might with our therapies, you will still get cancer at a higher rate than you would if you were younger. So you always have to treat it. You always have to have therapy that can go and fight it. And that therapy will not be necessarily a rejuvenation therapy. Over the Grave, for example, is very excited about this company called Maya. They are developing something called 6-thiodioxyguanine, which is a milder form of chemotherapy. We might need this as well for cancer. I guess that's my take there. That is not like there's no like if you could like wave a wand and make everything young in an organism except the DNA mutations. I don't think that will be enough to bring the mortality from cancer or incidence back to when you were younger, unless you could CRISPR everything. Like one could imagine this, right? You could do a highly multiplex CRISPR editing or make p53 functional again in every single cell. There are so many cells, so much that could go wrong. I think cancer happens.
There are some crazy ideas. I don't know if you've heard about like in vivo sequencing, where you do sequencing of cells in the person's body. You map the somatic mosaicism. Then you could actually, if you had a sequence-based way of killing cells, kill a cell only if it had a particular sequence. You could maybe start to manage the somatic mosaicism by selectively killing off lineages that are problematic.
That would one that would probably decrease the odds. Then you have more mutations happening between treatments. I think assuming that you will get cancer, and then being really good at predicting, screening, detecting, and killing, assuming it happens. I think to me it's the better approach. Because I think it's like once if you're like truly and like robustly cancer free for a while, some people can live many years without incidence of a different cancer. If you have this robust cancer cure, and then you have like a robust way of reducing heart disease, you can like do like if you screen your arteries, you're like if you're cancer free and you're heart disease free, and then you can improve. If you improve cognition, you can also do scanners for like a neurodegeneration. There are like many things you can show that you're rejuvenated, and if you have all of that information without a lifestyle trial, I think it will be reasonable to say that you will live a longer, healthier life. You could go from the causes and risks from falls, for example, like breaking a hip or something. If you have a stronger bone and stronger muscles, mobility is higher. If you have all of these things, it will be very unusual if your mortality from falls would not go down. It will be...
Right. This is the idea of surrogate biomarkers. I don't know, were you at the Biomarkers of Aging Symposium in December?
I wasn't. I had work to do here, so I was
there. But I think this discussion, one of the things that Kristen Fortney mentioned is that during their research process, they measure everything that they can. But when you do a clinical trial, the incentive is to measure less because you don't want to identify a side effect. And that's a bit of a perverse incentive that is actually a big problem for the aging field, because ideally what you want to do is when you're doing these trials, measure as much as you can, because the only way we're going to ultimately figure out what the best surrogate biomarkers are is by measuring them in the context of interventions. Do you guys have a strategy for that?
So I think there's sometimes discourse about aging biomarkers. I think that's somewhat mistaken. At the cellular level, you could say that DNA methylation is a great aging biomarker. Most of cellular aging is epigenetic aging. The more it looks like a young cell epigenetically, the better it is. At the organismal level, I think functional metrics are good enough. If you act like a young person in all these ways, you're probably fairly rejuvenated. I think we're not going to find any like weird like this one molecule and you're young now. If everything looks great functionally, then
you're probably... like your skin elasticity is good, your bone density is good.
Yeah, because the thing with aging is it's not like there's an aging molecule that goes up with age, and if you measure the molecule and make it go down, then you're healthier. It's many processes.
I wasn't implying that there would be one, but there might be some set of a few hundred that is...
Yeah, I mean, it's like you could build, let's say, you could build, for example, a proteomics or a kind of clock of some kind. I maybe prefer a proteomics clock than a methylation clock from blood, because that's just measuring PBMC, methylation, and their composition. Proteins are more unbiased sampling of what's happening in your body. But even then, the clock seems one step removed from the function. If you had this clock and it looks healthier, there's always but is your hip mobility better? Like, is your bone density? I mean, maybe it's cheaper to measure, but if you can afford to measure, there are things with a full physical assessment or a battery of cognitive tests. And in this case, you have to measure yourself not against a healthier person of your age, but a young person. What Brian Johnson is doing, actually, what he is doing is actually those are the biomarkers of aging. It's like measure everything in that way, and if that it all looks young, you have to measure against a healthy young person, not against an unhealthy 8-year-old.
Not like the healthy for your age thing. That's something
Yeah.
we need to get away from.
I think there's probably too much thinking about biomarkers of aging. I think it's good with what we have right now. Like, I think if you could truly rejuvenate someone, you would see it very easily. That's maybe my take. If you have a tiny effect, it's like statistics. If the effect is huge, you don't need statistics. You just see it. It's only when the effects are kind of like a hair splitting when you have to like do statistics and t-tests. Like, if someone's hair turns is white and turns black, you don't need to like compare the pixels and see that there's more pixels of black now. It's obvious. If you're chasing big effect sizes, identifying if it works becomes easier than if you're chasing like small effect sizes. Like if you decrease Alzheimer's progression by 10%, that's hard to measure. If you stop or reverse Alzheimer's, the patient's going to be like,
I've heard that Retro plans to have clinical trials in the single-digit years from now. I'm hoping those are big effect size therapies.
Yeah, we hope that too.
Seems to be what the goal is there.
Yeah, I think that many of the things now that we're thinking about trials is that, I come down here in the office, check out the experiment, see the mice. Sure, many things work great in the mice, but then famously there's the whole in mice meme. And then there are like a lot of thinking that we can do to make sure it would translate. But then there's always many people before us have been also have those thoughts and tried very hard and then they have failed. So until you put someone in, that's when things get real. All the careful thinking could be actually wrong because biology is something that it's like a surprise. It's going to be interesting when we actually do the first real. Well, it's like phases 1 are like very like not that interesting because it's just like safety, is the drug safe. I think that ours are fairly safe. Efficacy is like showing that the drug is actually doing what it should do in a human. That's where I think it's going to be a very tense moment, like make or break. We have funding to do many of these phase 2 trials, fortunately, but we'll see when we get there. Because that's where I guess kind of the before and after moment for many companies, where you're like in this fairyland of research where everything's great and you can do whatever you want to the mice. You can slice the brain, look at the brain, get the bone, measure anything you want, and iterate very fast. But at some point, you have to define the therapy, to make a GMP, put it in a human, and you have to stop the research, commit to something for a few months, get this thing working, fingers crossed that it works. And you might even just completely fail. And it's a huge commitment in money to try it.
All right. Final thoughts section. Who are the people that you follow? What are your information sources? Are there specific labs, podcasts, people that you follow on social media? Who are your most what sources of information do you like the most?
That's a good question. I use Feedly for RSS. I'm an RSS maximalist. I follow website-wise or aggregate lifespan.io and Fight Aging. They collect many interesting results that come out. I also follow journals. You can follow journals like Nature Mechanisms of Aging or Aging Research Reviews. There are a couple of aging journals. I think Aging Reviews also have a Twitter account. And then I think not that there's like any specific person. It's more like I follow 1,000 people on Twitter. I try to do everything that comes out. Sometimes I just search at work trying to figure out things to inform our programs, like why old people die more often of COVID, but vaccinated people don't. So what's going on, and how can we show that our therapy would or should we go for that as an indication? So in that process, I end up doing like extensive literature reviews to Google Scholars, starting from nothing. For example, I guess I follow most people you might have in mind that are kind of known in the field, but I don't necessarily subscribe to everything of the papers. Like I don't have like a Horvath alert, which is like Horvath paper, and no, it's like if the paper shows up in a journal, I might have a look if it's interesting. I guess I have a sense of taste for what I filter. So like there are so many things to read. It's like it's more like what do I choose to read? If someone has a like we tested this new Rapalog in dogs, I will completely ignore that. If someone has like a reprogramming paper, I will be very interested in reading it. Or if someone has a paper where they do a replacement, like we put a young liver in an old mouse, and then here's what happened. Or we do a muscle replacement, and what happened. Or we replace the entire gut. It's like, oh, wow, what's going on? Or we made the programming much more efficient. I guess things of that sort I tend to find more interesting. Editing papers, I don't tend to go so much like we improve CRISPR by X% and there's fewer off targets. We probably not read the paper usually. If someone has a computational paper for perturbation prediction or interesting methods, I will probably read it. to see what they're up to. Sometimes if papers also on things like making these things into how to scale, how to make things GMP, replacing components for like GMP friendly components. I guess it's very more like linked to our interests as a company to make things not just research grade but clinical grade or things. Sometimes read broad theory pieces, like, for example, João Pedro de Magalhães has his critique of the hallmarks paper, or we sometimes have some more like theory pieces. There's also someone I like, I guess, to put a name, Dan Enninger. He's a German PI, most famous for having his line is like that rapamycin is not an aging drug. They did the most comprehensive measurement of rapamycin effects in mice, showing that for many things it doesn't do anything, or other things it does, and for some other things it also works in young mice. It improves performance in young mice too, not only in old mice. It's a very interesting guy that I guess is maybe lesser known than the big names in the field.
Yeah, I hadn't heard that one. I'm going to look that one up. That's a good one.
Yeah, I just want to see stuff in my latest post on my blog. Because otherwise, I think I'll listen to any aging-specific podcasts.
What's your blog, by the way, for our listeners?
Oh, yes, my blog is nintil.com. That's nintil.com.
Okay.
But yeah.
And you mentioned fightaging.org, which is another, I think, a very good aggregator of information.
It's like the author, he really, like, there's so much stuff in there. Or like, even both the article, but also as starting points to go deeper into like into more things, or like bring things to your awareness that maybe you didn't know you wanted to know about. Of course, there's so much stuff in that website. You have to filter for what you are interested in. If I go now to Fight Aging, what do we find here?
Yeah, he has a good year in review article out right now that I've only halfway read it.
Yeah, for example, if there's like, is alternative splicing a meaningful cause of aging or a side effect? That one I might usually ignore. Papers that try to tease out some causality questions that have the wrong lens, I usually ignore, because everything is connected to everything else. I'm like, we already know that. If it's something careful, if it's something experimental, trying to do causality work, that I will read. I think on that, the paper from Ken Raj was quite good, where they're like, okay, if you boost mitochondrial health, does that affect epigenetic aging? If you do something to telomeres, does that affect epigenetic aging? Little tweaks to the hallmarks, and then see how that careful work I tend to like. Someone should do all the combinations. Like, if you poke at this thing, does it affect your thing? They should do all the combinations of like, poke this hallmark, affect your hallmark. That paper will be quite cool to read. I hope someone does it someday.
And then I guess we should always ask, how could our listeners help you? Like, what does Retro need? What are you guys looking for? What would help you guys make faster progress?
Yeah, so we are hiring all the time. We currently have a couple of positions open in our HSC team, and automation as well. Maybe the ones we are hiring the most right now, it's automation, which is both people with experience in lab automation, working with automation, but also software. We do think that good software engineers are usually very open-minded, smart people that can learn very fast. So if someone has a good foundation in software and can work with things like Python, they can probably learn lab automation here at Retro. I think we might actually prefer someone that's a good software engineer than someone that's a good automation engineer, because it's maybe, I don't know, there are probably more software engineers than good automation engineers. And if you're willing to work on aging and basically go from 40 experiments to 4,000, if that sounds exciting, if they find the idea of someone very smart doing pipetting repulsive and want to uplift mankind into the era of automation, they should definitely come our way. And either email me or email Ben Ray. My address is Jose@retro.bio. Ben is Ben@retro.bio. If they want to come our way to work on these problems. If you're interested in perturbation prediction, email in bio for rejuvenation. Also, they could be welcome to talk to us. And yeah, anyone working on things like reprogramming, ex vivo reprogramming, in vivo reprogramming, chemical reprogramming, gene circuits, that's definitely up our alley. So they should also come our way. Either, yeah, it's like even if they don't see anything on our website that like it's a perfect match for them, just email me and then we kind of have a conversation. Oh, saying you're muted.
Nathan, anything else?
No, I mean, that was really great. So
thank you so much for your time, Jose. Retro is one of our favorite companies in the space. We're very excited to see what results come. We will keep trying to send you awesome people. And hopefully listeners of this podcast that aren't even LBF members will reach out to you guys and maybe be able to give you a hand and accelerate your progress.
That'll be
because
amazing.
that is our podcast, the Longevity Acceleration Podcast.
Yep.
So we want to accelerate everybody.
Longevity hack, as I would say on Twitter.